Hello, and today we're going to be doing some DNA replication modeling. We're going to start by making a DNA strand, then we'll go through the replication process using that strand. So we're going to do that using the licorice, some toothpicks, and these marshmallows we have here. We're going to start by creating our DNA sequence, our DNA strand. We have the sequence here, GACTCA, on this page, so that's the sequence we'll use, and we have a key. So in green is going to be guanine, yellow is going to be cytosine, pink is going to be adenine, and orange is going to be thymine. So we're going to start by just taking a toothpick and putting it into this piece of licorice. This licorice is going to represent our backbone, our phosphate, and our deoxyribose, making the backbone of our DNA structure. Then in the middle, we're going to have our nucleotides, G, A, C, and T are going to be our different nucleotides, and they're going to go on this toothpick and be represented by the different color marshmallows. So starting at the beginning, we're going to have a green marshmallows for guanine, for G. We'll slide that all the way down, and then every time G, hopefully we know by now, G always pairs up with C, so we find C cytosine, and that's going to be a yellow marshmallow, and we'll put that on that same toothpick, and that goes right next to it. Then we'll keep working down our sequence, so then we'll go to our next one, and as we work down, our next one is an A. We look on our key, it says that's going to be a pink one, so we put on our pink marshmallow. Again, we slide it all the way down, and now on the opposite side of A is going to be T, because A and T always pairs together. So look on our key, we see thymine, and we're going to put that onto our toothpick. We'll keep working our way down to our next one. So our next letter is going to be a C. So I'll put my toothpick on, and then I look on my key and I see that's going to be a yellow, the yellow marshmallow for C. And then again, C always pairs up with G, so I'm going to get my green for guanine for G and place that on. Then we go to our next sequence and we have a T. So we put our toothpick on. Again, we look at our key. T is going to be orange, so we put our orange marshmallow on. And again, slide that down, pair up. It's going to pair up with an A or a pink marshmallow adenine. And then next is a C. So I do a new toothpick on here. C again is going to be yellow, so we get a yellow marshmallow, slide that down, and then we go to our next one. Our next one is going to be, well, we got to pair that one up, so we got to get our green guanine to pair up with our C cytosine. Then our final one is going to be an A, so I take my last toothpick, I'm towards the bottom of my licorice strand, and I put my A on. That gives us our DNA strand once we pair up our A with our T, our orange thymine. So now we have our DNA strand. However, it's double stranded. We want to make sure we have the backbone, that phosphate and deoxyribose on the other side. So we're going to take this and we're going to add on a piece of licorice to the other side to give us that full DNA structure. So that gives us our DNA model, and from there we'll be able to do the replication process once we have that model complete. So once we have this model, if you look at it, you can see that ladder shape, that ladder structure, and then if you hold it from the top and bottom, we can twist it and we can see that double helix shape you're familiar with for DNA. So that gives us that double helix structure for DNA. So that's our DNA model. From there, we'll go through the replication process next. So to show the replication model, first we're just going to mark each one of these marshmallows so we know which ones are part of our original one and which ones are new nucleotides we're adding into our model. So I'm just using a blue marker because none of my marshmallows are blue. I'm using that to indicate and mark which ones are part of my old strand. So I have each of my marshmallows marked, and now we're going to go through the DNA replication process. So the first step is DNA helicase is going to come through and unzip this DNA. We're going to represent that by first taking our licorice piece that we added on one side off. So that's starting the unzipping process. And in real life, it would zip down the middle. We don't want to break all these toothpicks, so we're not going to zip down the middle. But instead, we're going to add toothpicks to this piece of licorice that we just removed. So we had 6 nucleotides, 6 base pairs. So I'm going to do 6 toothpicks into this piece of licorice here. I'll move this one off to the side out of my way for just a second, and then we'll pull that back over once we got all 6 of these toothpicks in. I got all 6 in. Now what I'm going to do is I'm just going to take the marshmallows from the right hand side of this model, so 1 pair from each one, and move it over to this toothpick on my second DNA strand. So I'm just going to take 1 and slide it over and then slide it down, just like we did at the very beginning of this process when we made our original model. So we take that and slide it all the way down. And I'll do that for all 6 nucleotides, and that gives us 2 single-stranded DNA once we're complete. And again, this process is completed by the enzyme DNA helicase. So helicase is coming through, unzipping that DNA or unzipping the genes, so we can have single-stranded DNA. That single-stranded DNA, and then it's going to move on to our next step. So we have single-stranded DNA, 2 single-stranded DNA. I had 1 fall off as I went through that process. I'll make sure that stays on. So now I have 2 single-stranded DNA. Next, what we have is DNA polymerase. DNA polymerase is then going to run through, and I'm going to put these in the order we started. DNA then is going to run through and add in our new nucleotides. So we have 2 single-stranded. To add in those new nucleotides, we got to pair up and match up what these base pairs go with. So if I had a yellow on the top, that's our cytosine, I'm going to put a green to pair up with it. So G and C still go together. So I still have my same base pairings, and I'm going to go through all the way and pair them up. So again, my orange is thymine. I pair that up with A, adenine, and work my way down. Next, I have a green for guanine, so I pair up with C, which is our yellow one. Pink one, which is A, I'm going to put a T, which is our orange. Another green one, so I'm going to go with a yellow for cytosine, and at the end we have T, which pairs up with A. And we do the same thing on our second strand. Typically, DNA polymerase works from a 5' to 3' direction. So if it comes down on one, it would actually start from the bottom and work up on the other one. So I'm going to do that same process over here, starting from the bottom. So I'm going to start with A and pair up with T. Work our way up. We have a C. I'm going to pair that up with my G. I have another T. So now I'm going to pair up with A. Then I have my C. I pair up with G. I have an A, so I need to pair up with T. And finally, I have a G, so I'm going to pair up with C. So this is DNA polymerase adding in those new nucleotide base pairs. However, I need to put those nucleotides together and close off my DNA sequence. I'm going to do that by adding this piece of licorice. The enzyme that actually connects things together, closes off our DNA, is going to be DNA ligase. So this isn't exactly how DNA ligase, but the final one to close off our DNA, we would be looking at DNA ligase to close it off. So I'm going to again close these off, same way as we did earlier, just close it off and connect my DNA strand and create that double helix structure. So I got my first one connected and it's all complete, and then I'll do the same thing on my second one. I connect it and make sure everything's hooked up correctly. Once we have them all connected, then we'll look at our marking and kind of see what happens. So first, we got them all connected. In this case, it's called DNA replication, so we want to make sure we have 2 identical copies. Sometimes you're moving them around, you get them flipped around, and it doesn't look identical like right now. So in order to do that, you may have to rearrange things or rotate things, and then you can see that we have identical. It goes green, pink, yellow, orange, yellow, pink, and then the corresponding matching pair on the opposite side. So we have those matching pairs. Then I'm going to rotate these so we can see the marked ones. And if you notice, all our marked nucleotides are on the same side of the strand. They're all on this side. All the blue markings, if we look at them, are all on that left-hand side. And then the right-hand side has no marked ones. If I rotate them around, you can see it's not marked. So it's all marked on the same side. And then the same thing's true on this one. All my marked ones are on the same side. In this case, the right-hand side of this DNA strand. So we have them all on the same side. We call that a semi-conservative process. So one side's all the same, it's all from the original, and the other side's all new nucleotides that were added on. So that's that semi-conservative. Each new strand has half from the original strand and half new nucleotides. And then we have our DNA replication, because we have 2 of these DNA double helix, double-stranded DNA structures. They have an identical sequence. However, we made a copy, half the old one, half the new one. It's that semi-conservative process. So hopefully this helps you with the DNA replication process. Again, our three major enzymes we looked at today was helicase, unzipping it, polymerase, adding in our nucleotides, and then ligase, connecting those nucleotides together, closing it off. Thank you. Hopefully this helps you with DNA replication.